Hey guys, welcome to chapter 17.3. We are still talking about gene expression, but more specifically in this section, RNA processing. So what we are doing is we are looking at the nucleus, and last time we talked about transcription, which was taking the information from the DNA and transcribing it into a pre-mRNA strand. And what we're doing now is we are going through RNA processing. We're going to take some stuff out, we're going to add some stuff, and we're going to make the messenger RNA, or the primary transcript, which will exit the nucleus. And let's start at the beginning and then move to the end, and then we'll talk about the middle later. So as RNA polymerase finished adding the first 20 to 40 nucleotides during transcription, remember that it was working at the 5', it started at the 5' end and worked to the 3' end. So it reads up and it writes down, so it's writing down. So as it did the first 20 to 40 nucleotides, a 5' cap was added. So this 5' cap is a modified form of guanine. A G right here is added. And what is it attached to? It is attached to, you can see this is weird, it has phosphates. Why do we see phosphates? Well, remember that RNA is made of ATPs. It is a triphosphate. And so while they're hydrolyzed to fuel the process of transcription, this first one was not hydrolyzed. So you can see that we have still we have the whole molecule here. It's the pyrophosphate was not removed. So we have the full ATP on the mRNA strand. And so that modified guanine is added to the phosphate. And that is going to function to protect the strand from being degraded or hydrolyzed by enzymes. There are enzymes in the nucleus that function purely to break down nucleic acids. These are called exonucleases. And they're not always very specific. Sometimes they just find nucleic acids and start degrading them. And so this is going to protect the message. The exonuclease won't be able to grab on. And now we're talking about the other end, the poly A tail. Let me move this guy up a little bit. Here we go. The poly A tail. Remember that during transcription, the RNA polymerase II would reach a polyadenylation signal. Remember that it would get to the end of the message in a eukaryotic cell, and it would hit AAU, AAA. Remember that it's not universal, but we're going to use that as our example. And then when it reached that polyadenylation signal, it would jump ahead 10 to 35 nucleotides. And so what we're doing now, we're going to modify this mRNA strand, and enzymes are going to move towards that polyadenylation signal, and it's going to start adding adenosines, many adenosines, 50 to 250 adenosines, nucleotides, are going to be added to the 3' end. And so we call this a poly A tail, poly meaning many, A meaning A, or adenosine, and tail meaning the end. The poly A tail has a few different functions. The first one is that it seems to facilitate the movement of the primary transcript out of the nucleus. So it's going to be the molecule that is able to leave, or the message that is able to leave the nucleus. And it is helpful because this poly A tail is there. It also is going to protect the mRNA, once again, from those exonucleases. It's not going to allow them to sort of latch on and degrade the message. Finally, it's going to help the ribosome or sort of direct the ribosome to attach to the 5' end. So we know that the ribosome is going to need to attach to the 5' end in order to start pumping up the message of the protein or the amino acids. The 5' cap and poly A tail will not be translated into the protein. However, these are not the UTRs, or the untranslated sections, or the untranslated regions. So there's two untranslated regions on your primary transcript. So right here we have our 5' cap, or the guanosine, and the triphosphate. And then we have the untranslated region right here, and that's going to stop right here at the start codon. So the spot where the RNA polymerase II is going to start adding nucleotides. So this is untranslated, and we're going to talk about this later on in future sections. And the other untranslated section does not include the poly A tail. It is essentially the section of the polyadenylation signal here. And so that is untranslated as well. So they call these UTRs, the 5' UTR and the 3' UTR. RNA splicing occurs when large portions of the RNA molecules are removed. And it's a little counterintuitive. So we see here, we have introns, and we have the red segments are called exons. And it would only make sense if the introns stayed in, but they don't. The introns are what are removed because introns stands for intervening sequences. The introns are going to be taken out, and the exons are going to be glued together. So we can see here these red, or spliced together is a better word. So you can see these red spots, so the exons are spliced together, and this is the message that is going to be coded. So the introns, while they're very important, are not included in the message or the final transcript. The coding segments of nucleic acids are called exons because they're going to exit and because they will be expressed. That's how I remember it. So the exons are going to exit and then be expressed. They are going to be spliced together by a really cool little enzyme called a spliceosome. The spliceosome is a complex of proteins and RNAs. So RNA is not only messenger, it also can function as an enzyme. And so spliceosomes not only assemble the exons together, they also catalyze the splicing reaction. And so it was discovered when scientists discovered ribozymes, and ribozymes are RNA molecules that function as enzymes. And RNA is an ancient molecule that exists in both bacteria and in eukaryotic cells, and it can function to catalyze reactions. So it lends itself for us to consider if ribozymes are the ancestor to proteins. Just something interesting. And what's pretty cool, in some organisms, the pre-mRNA splicing process is going to happen even without the aid of any proteins. The mRNA or the exons are going to sort of splice themselves together because that RNA can function as an enzyme. And so the take-home message here is that not all enzymes are proteins. RNA functions also as an enzyme. And so how does the splicing thing happen? Let me grab the drawing tool, and I'm going to draw some pre-mRNA, and I'm going to make a couple of exons. I'm going to make 3 exons. So here's an exon, and then that's an intron. Here's an exon, and here's an exon. And so what the spliceosomes do is let me erase this. Boop, boop. They're going to take the mRNA, and they sort of it's going to fold up like this, and let me make this longer. The exons are going to be lined up here and here, and here's our exon number 3. And the spliceosomes, let's color those red, are little enzymes, RNA and proteins together, that are going to splice, and they're going to cut and reconnect these exons together. And that is the job of the spliceosome. And it is, like I said before, it is not only a not only is it proteins, but it's also RNA. So that's pretty interesting. So ribozymes, there are 3 properties about RNA that allow it to function as an enzyme. First thing, remember that RNA is single-stranded. And so this is going to allow for it to base pair with itself. So we can see in this picture here, this strand of RNA has a 3D structure. And that's because if you think about protein folding, the intermolecular forces that allow for the protein to fold can be present in here too. So we have the hydrogen bonds between the nucleotides that are going to allow it to base pair and to give it this 3D structure. A second aspect to enzymes that allow it to function as an enzyme, to RNA that allows it to function as an enzyme, is that it's going to also be able to contain functional groups. So where proteins have amino acids, these enzymes or these RNAs can contain functional groups, which will give it more catalyzing ability. Third, RNA's ability to base pair is going to allow it to find other nucleic acids. So if it's looking for a piece of DNA, a section of DNA, and it'll be able to find a very specific sequence and to line up to it because it also contains that sequence or its complementary base pair. And so it's going to be able to locate very specifically spots of DNA so that it can begin a reaction or it can complete something. It can complete a process. So those are the reasons why ribozymes or why enzymes can function, or why RNA can function as an enzyme. Let's now talk about the importance of introns. So these are the sections that are going to be removed. And it seems interesting or confusing to think that they're going to be of value if we're just going to remove them. Let me move this up so I've got some space to, it's too high. So I've got some space. Oh well. If we're removing them, it seems like it would, that they'd just be junk DNA. However, it allows for alternative RNA splicing. It allows for each gene to have multiple, or to be able to code for multiple proteins. Let me explain to you what I mean. So let me draw our enzyme again, or our, RNA, pre-mRNA strand. And we're going to draw out the 3 exons again, 1, 2, 3. And last time we folded and we connected these 2 exons. Let's actually put a 4th one in here too. Boom. What this allows, alternative splicing allows, is for if this molecule of pre-mRNA were to fold in a different way, these spliceosomes could connect. Here's exon 1, exon 2, exon 3, exon 4. So it's going to connect, it's going to leave in these exons and it's going to connect these exons. And that's going to create a very different message. And this is going to allow each gene to create not just one protein, but many different proteins, because it's going to include different pieces and put different pieces together. And this explains why a nematode can have the same number of chromosomes as a human, while humans have so much more complexity, because our genes can be expressed in a greater number of ways. And so secondly, the proteins are composed of domains. So introns are going to function here. What it's going to do is it's going to give, let me use the black color. It's going to put more space. If we look at this polypeptide, this is a polypeptide, this whole thing, and it's going to have 3 different domains. So one strand of amino acids, and it's going to put space one strand of mRNA. It's going to put space between these domains. And this domain is going to code for a specific section on a protein. And if we move these sections a little bit further apart, when crossing over happens, it's going to allow for a greater chance that this whole domain is going to be able to move as one whole section, or this domain can move as one whole section, so that when it recombines with the other chromosome, its homologue, it's going to recombine, and it's more likely to be able to function, more likely to be able to be expressed, because it's going to have, it's going to be more intact. And this is actually a great way for variation to occur. This is going to allow proteins to be expressed in different ways if recombination happens and this domain is altered a little bit and it's recombined with the rest of a protein. It's going to allow for that trait to be expressed just a little bit differently. All right, so that is 17.3. I hope that helped, and let me know if you have any questions.